If you look at the difference between metastasis and invasion, metastasis is the spread of tumor that moves from the site of origin to a site which is discontinuous with the primary tumor. Whereas if you look at invasion, the invasion is the direct extension and penetration of the tumor cells to the neighboring structure. So the invasion is in continuous alignment, whereas metastasis is the spread of tumor which is not continuous. In order for the tumors to grow, the tumors need to do two things. One, it should be able to survive against the host immune system, and number two is that it should be able to survive in the newer host environment. Once both the criteria is satisfied, the tumor starts to move, and the first step is called as the local invasion of the extracellular matrix. The extracellular matrix is formed by the basement membrane, the interstitium, and the basement membrane of the blood vessels. The tumor cells then starts to move inside the lymphatics or the blood vessel, which is called as the intravasation, where it has interaction with the cells like the lymphocytes and the platelets. Then the tumor cells with the along with the platelets starts to move inside the blood vessels, forming tumor cell embolus. Once the tumor cell embolus finds the perfect environment for it to survive, uh, it comes out of the blood vessels, which is called as the extravasation, and forms small clump of the tumor cells as a micrometastasis. But then eventually the micrometastasis starts to grow, gets proliferate, and gets bigger over the near site, uh, forming a bigger clump, which is now the macroscopic tumor. So the metastatic cascade is divided into two phases. One is the invasion of the extracellular matrix, and the second part is the vascular dissemination, and then comes the homing of the cells. The invasion of extracellular matrix can again be divided into four different types. One is the loosening up of the tumor cells and the tumor cell interaction. Then we have degradation of the extracellular matrix and attachment to remodeled extracellular matrix components, migration and invasion of the tumor cells. Uh, Next comes the topic, which is the loosening of the intercellular connections between tumor cells. So it usually begins with the epithelial mesenchymal transition in cases of some cancers, the carcinomas, which means that there is downregulation of the epithelial component and the upregulation of the mesenchymal component. Now, the tumor cells are usually held together by a connection which is called as the E-cadherin. Now, these E-cadherin have the cytoplasmic portions which is called as the beta-catenin. Now, until and unless the tumor cells, the tumor cells have the breakage of these intercellular connections, that is the E-cadherin, the tumor cells cannot invade or the tumor cells cannot metastasize. Therefore, the tumor cells, in order to metastasize, must have the uh, inactivation of the E-cadherin, which occurs by the epithelial mesenchymal transition, the mutational inactivation of the E-cadherin gene, or the activation of the beta-catenin uh, genes, or if there is inappropriate expression of the two transcription factors, namely the snail and the twist transcription factors. So all these elements help in the inactivation of the E-cadherin and thus helps in the process of invasion and metastasize. Uh, Now we have two changes. One is there is a loss of the epithelial cell receptors like integrins. Now, once there is loss of the epithelial cells, the receptors, the integrins, there won't be connection between the collagen or the laminin and uh, the epithelial cell receptors, the integrin. So loss of this interaction between this collagen or laminin with the integrins is going to give rise to two different situations. One is the loss of the resting state of the tumor cells and the loss of differentiation of the tumor cells. Second, we have the modification of the matrix. Modification of the matrix is also going to help in the process of invasion and metastasis. If the tumor cells need to penetrate the or go through the extracellular matrix, the tumor cells should be able to digest the chief component of the extracellular matrix, namely the collagens, the glycoproteins, and the proteoglycans. So all of these, the main components of the extracellular matrix, are proteins. So the enzymes that are able to digest these proteins are called as protease. These proteases are matrix metalloproteinase, cathepsin D, and urokinase plasminogen activator. Some of these proteases, like matrix metalloproteinase, not just helps in digesting the extracellular proteins, matrix proteins, but also helps in the processes like the release of the sequestered growth factors, which helps in the proliferation of the tumor cells, and the chemotaxis that helps the tumor cells to move towards the newer site, uh, and also helps in process of angiogenesis, because if the tumor needs to proliferate, uh, the they should have the proper blood supply and the nutrients, for which there should be proliferation of newer blood vessels, which is also enabled by matrix metalloproteinase and also results in the growth promoting effects, which helps in the tumor cells to survive and grow in the newer host environment. The process of movement of the tumor cells is called as the chemotaxis and or locomotion. Now the movement is assisted by the protein inside the tumor cells. These proteins are called as actin. So the actin helps in the process of chemotaxis or movement or locomotion. The process of chemotaxis is assisted by two different activities. One is the autocrine and the other one is the paracrine effect. So the process of autocrine is because we use the word autocrine because these motility factors are produced by the tumor itself, uh, and the word paracrine because in this condition the stromal cells produce the several factors like the hepatocyte growth factor and the scatter factor, which helps in the process of chemotaxis or the locomotion. Uh, in addition to these factors, we also have several other factors that help in the process of locomotion or the chemotaxis, which are the cleavage products of matrix components. For example, the collagen or the laminin, and the growth factors, for example, insulin like growth factors 1 and 2. Now comes the step where the tumor cells enter inside the blood vessels. The tumor cells, once inside the blood vessels, have the interaction amongst each other, which is called as the homotypic interaction, and the tumor cells will also start to bind with the platelets. Now, this interaction is called as the heterotypic interaction. Also, they can have the interaction with the lymphocytes via the receptors present over the surface of the lymphocytes, which are the CD44 expression. Other factors could also help in the better survival chances of the tumor cells inside the vasculature. Uh, for example, there could be production of fibrin around the tumor cells, and second could be the things like plasticity, that is the tumor cells, they develop the stem cell like property that will help in the process of better survival and the proliferation of the tumor cells. And now comes the process of extravasation, where the tumor cells will start to move outside the blood vessels and start to proliferate in the newer host environment. Uh, Now, for the tumor cells to survive in the new metastatic site, these tumor cells need assistance by some of the cytokines, the production of growth factors, and the production of proteases. This concludes our metastatic cascade. Thank you.